Students, today we are going to discuss test for molecular basis of inheritance. Question number 1. We have to check the correct statement. DNA serves as genetic material in most of the organism, which is true, and RNA primary function as messenger RNA. That is also correct. RNA also act as genetic material in some of the eukaryote. It would be some viruses, not eukaryotes. So this statement is incorrect. The process of transcription involves synthesis of RNA from DNA. That is absolutely correct. DNA to RNA is transcription and RNA to protein is translation. Human genome sequencing project has set in a new era in genomics. This statement is also correct. You can check all the statements in NCERT also. So statement 1 and 3 and 4 are correct. Statement 2 is incorrect. So option A is correct. DNA and RNA differ by, if we consider the structure of DNA and RNA, then pentose sugar and nitrogenous bases are different in DNA and RNA. Sugar in DNA is deoxyribose sugar and sugar in RNA is ribose sugar. If we consider nitrogenous bases, then in DNA we have 4 nitrogenous bases, that is adenine, thymine, cytosine and guanine. While in RNA we have adenine, cytosine, guanine and uracil. So the difference is nitrogenous base and sugar. If we consider other option like nitrogenous and phosphate group is incorrect. Carbon atom in sugar, both the sugar have 5 carbons, so this is also incorrect. And sugar and phosphate, again phosphate group is incorrect. So answer will be A. Next question, identify the correct feature of DNA. It is made up of 2 polynucleotide chain. This is correct. Backbone is constituted by sugar and nitrogenous base. Nitrogenous bases are projected inwards. Backbone is made up of sugar and phosphate group. So this statement is incorrect. 2 chains have parallel polarity. 2 chains have anti-parallel polarity. So that is also incorrect. Bases in 2 strands are paired through hydrogen bond. This is correct. If we consider adenine and thymine, there are 2 hydrogen bonds. Cytosine and guanine, there are 3 hydrogen bonds. The 2 chains are coiled in left-handed fashion. This is also incorrect. It is in right-handed fashion. So answer will be 1, 4 are correct while other are incorrect. A double-stranded DNA of a particular species was analyzed and 17% is adenine. If adenine is 17%, then thymine would be 17%. Cytosine and guanine. Amount of cytosine will be 33% and guanine is also 33%. So the answer will be C. Amount of percentage of cytosine is 33%. Next question, statement 1. In prokaryote, DNA is held with some protein in a region called nucleoid. This is correct. If we consider DNA packaging, then in prokaryotes, DNA is packaged with the help of some proteins, and these proteins are positively charged, but these are not histone proteins. So the first statement is correct. The DNA in nucleoid is organized in large loop by some proteins, and these proteins are positively charged proteins. So both the statements are correct. Answer will be C. Both statement 1 and 2 are correct. Next question, the eukaryotic genome differ from prokaryotic genome. If we consider the difference between eukaryotic and prokaryotic DNA, then the DNA organization is much more complex with histone proteins in eukaryotes, while in prokaryote it is not that much complex. So answer is B. Next is diagram of nucleosome. So 1 is, this structure is DNA. This is histone octamer. So 3 will be histone octamer and 2 is H1 protein. So the answer is A. 1 is DNA, 2 is H1 histone and 3 is histone octamer. Next, the identification of transforming principle proved that. Now what is transforming principle? Transforming principle was performed by Griffith, and he used Streptococcus pneumonia, which had 2 strains, S strain and R strain. S strain was virulent one and R strain was non-virulent one. So in transforming principle, dead bacteria lethal as live one. This is incorrect. Streptococcus pneumonia evolved from pneumococcus. It is also incorrect. Genetic information is contained in RNA. Actually, transforming principle was performed to identify that what is the exact genetic material, and genetic material is DNA. That was confirmed by transform. That experiment was performed to explain that only. But unequivocal proof that DNA is the genetic material, that was provided by Hershey and Chase. So answer is C. Genetic material is contained in DNA. Pick the incorrect statement with respect to the criteria for a molecule that can act as genetic material. If we consider properties of genetic material, then it should be able to replicate. That is correct. It should be stable chemically and structurally. That is also correct. It should not provide any scope for mutation. That is incorrect. It should provide scope for slow mutation. So answer is C. Next, read the following statement and we have to find incorrect statement. The presence of adenine at the place of uracil also confers additional stability to the DNA. If we consider the statement, then there should be thymine. The presence of thymine at the place of uracil also confers additional stability to the DNA. So this statement is incorrect. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. That is why it should be stable enough, and that is why DNA is the genetic material in most of the organism. So this statement is correct. Both DNA and RNA are able to mutate. This statement is also correct. DNA can independently perform synthesis of protein. DNA depends on RNA for synthesis of protein. First, DNA has to convert to RNA, then RNA can synthesize protein. So the statement is incorrect. So the incorrect statement is A and D. A and D are incorrect. Answer is C. Next question, assertion and reason. Assertion is in eukaryote, the replication of DNA take place during S phase of the cycle. If we consider the stages of cell cycle, then we have three stages in interphase, G1, S and G2. S phase is for synthesis of DNA and duplication of centriole. So the statement is absolutely correct. The replication of DNA and the cell division cycle should be highly coordinated, because if it is not happening, then the cell will have a polyploidy condition. So that statement is also correct, but it is not the correct explanation. So answer is D. If you group bacteria in heavy nitrogen means N15, and then switch them to light nitrogen. So the medium is NH4Cl, ammonium chloride, and if we switch the bacterium to N14 medium, after switching you have equal ratio. We have to find light and hybrid ratio, ratio for light and hybrid DNA. So suppose we have this N15 DNA, both the strands are N15. After first generation we have two DNA, one strand is heavy and the other strand is light, right? In the next generation we have 4 DNA. One strand is heavy, another is light, and these two strands would act as template, and these are newly synthesized strands. So in first generation 100% DNA are hybrid, and in this 50% are hybrid while 50% are light. So the equal ratio can be seen after the second generation. So answer is C. Deoxyribonucleoside triphosphate serve as dual purpose. What are the purpose? These provide energy for polymerization, yes, and act as substrate. Both the functions are performed by deoxyribonucleoside triphosphate. Identify the correct statement from the following. Monocystronic eukaryotic structural gene have interrupted coding sequence. If we consider monocystronic and polycystronic structural gene is present in prokaryote, monocystronic structural gene is present in eukaryote. And monocystronic structural gene of eukaryote is interrupted by some coding sequence is interrupted by a non-coding sequence, and coding sequences are termed as exon, non-coding sequences are termed as introns, and introns are removed by the process of splicing. So monocystonic eukaryotic structural gene have interrupted coding sequence, yes, and those interruptions are introns. So this is correct. And the other options, if we consider polycystonic prokaryote, that is correct, but they do not have any interrupted coding sequence, it is incorrect. Monocystonic prokaryotic, that is incorrect, and polycystonic eukaryotic is also incorrect. So the answer is A only. Now RNA polymerase 1, we have three type of RNA polymerases in eukaryotes. RNA polymerase 1 catalyze synthesis of 28S, 18S and 5.8S RNA. Cistron is the segment of DNA which code for a polypeptide, and DNA ligase join the fragment of DNA. Promoter is the binding site of RNA polymerase. So the answer will be A, 4312. Select the correct statement from the following with respect to transcription. The RNA polymerase is only capable of catalyzing the process of elongation. That is correct. The coding sequences, if you consider it for like prokaryote, then RNA polymerase alone can only catalyze the process of elongation. The coding sequences or expressed sequences are defined as intron. Introns are intervening segments, are not express sequence. Express sequence are exons. So this statement is incorrect. Intervening sequence do appear in processed RNA. Intervening sequences are removed in the process of splicing. So this statement is incorrect. In bacteria, translation can begin much before the mRNA is fully transcribed. This is correct. That is why in bacteria, transcription and translation are coupled reaction. So the correct statement is A and D. Next assertion, sometimes the regulatory sequences are loosely defined as regulatory gene even though these sequences do not code for any protein or RNA. That statement is correct. Inheritance of character is also affected. If we are given it is not affected by promoter and regulatory sequence, this statement is absolutely incorrect. Inheritance of character is also affected by promoter and regulatory sequences because these sequences regulate the functioning of structural gene. So the answer is, assertion is true and reason is false. A gene is defined as the functional unit of inheritance. That is absolutely correct. The DNA sequences coding for tRNA or RNA molecule is also a gene. That is also correct. A gene can synthesize tRNA, mRNA and RNA. All the types of RNA is synthesized by a gene, but the mRNA contains information for synthesis of protein. So both the statements are correct. Answer will be C. Both statement 1 and statement 2 are correct. With regard to mature RNA in eukaryote, if we consider mature RNA, that means the final RNA which is formed after splicing. So in immature RNA or hnRNA, we have two sequences, exons and introns. Exons are expressed or coding sequences, introns are intervening sequences. So in mature RNA, introns are removed by the process of splicing and only exons appear. Exons appear but introns do not appear in the mature RNA. That is correct. Next, select the correct statement from the following regarding the transcription. Association with some factor after the specificity, alter the specificity of RNA polymerase to either initiate or terminate transcription. If we consider RNA polymerase catalyze only elongation process. For initiation and for termination, they require some factor. Like rho factor is required for termination and sigma factor is required for initiation, which alter the specificity of RNA polymerase. So this statement is correct. Terminator is located upstream of the structural gene. If we consider upstream location, then promoter is located. So this statement is incorrect. The transcription and translation cannot be coupled. We have already discussed that transcription and translation is coupled in bacteria. So this statement is incorrect. The mRNA does not require any processing to become active in bacteria. That is absolutely correct. In bacteria, mRNA does not require any processing. That is why transcription and translation is coupled. So option A and D are correct. Read the given below statements carefully. Chemical method was developed by Hargobind Khurana for synthesis of homopolymer and copolymer of RNA and not DNA. So the statement is incorrect. Several enzyme was helpful in polymerizing DNA with defined sequences. That is correct. The process hnRNA contains only exon in eukaryote. We have already discussed hnRNA is inactive RNA which contain both but processed hnRNA is mRNA. So mRNA contains only exon. That statement is correct. Marshall Nirenberg made a cell-free system of translation for protein synthesis. That statement is also correct. So answer is B. Only B, C and D are correct. Which of the following statements about RNA polymerase are correct? RNA polymerase of bacteria transcribe all types of RNA, tRNA, mRNA and rRNA. But if we consider eukaryotic DNA, then we have at least 3 types of RNA polymerases. RNA polymerase is 1 and 2 and 3. Second statement is RNA polymerase is to transcribe snRNA. This is incorrect. It actually transcribes hnRNA. RNA polymerase 3 transcribe hnRNA because RNA polymerase 2 transcribe hnRNA. So this statement is incorrect. RNA polymerase 2 transcribe hnRNA. That is correct. So the answer is A and D are correct. All strains of Streptococcus pneumonia are virulent. It is incorrect. We have just discussed that Streptococcus pneumonia has 2 strains S and R, in which a strain is virulent one. A strain of Streptococcus pneumonia produce smooth colony. Yes, that is correct. So the answer will be statement 1 is incorrect and statement 2 is correct. In some viruses, RNA is present instead of DNA, indicating that RNA can also transfer heredity and it can act as genetic material. Next is match the column. If we consider Erwin Chargaff, then Erwin Chargaff gave base pairing between 2 nucleotide chain. Frederick Mesher gave the term nuclein. Francis Crick gave central dogma. Every McLeod-McCarthy gave biochemical characterization of transforming principle. Transforming principle was given by Griffith, but biochemical characterization was given by these 3 scientists. So answer will be A. Next is assertion reason. The split gene arrangement probably represent ancient feature of genome. This is correct and it is very important statement of this chapter. And the presence of exons is reminiscent of antiquity. That statement is incorrect because it should be the presence of introns and not exons. So answer is assertion is true and reason is false. Both the statement, this question is very important. So just refer NCERT for these 2 statements. Next is how many statements are correct. The process of copying genetic material from one strand of DNA into RNA is termed as transcription. The DNA dependent RNA polymerase catalyzes polymerization in only one direction. If we consider RNA polymerase or DNA polymerase, both the enzyme catalyze polymerization in one direction, that is 5' to 3'. That is correct. The promoter is located toward 5' end upstream and of the structural gene. That is correct. We have just discussed this point. The terminator usually defines the end of transcription process. That is also correct. So all the statements are correct. 4 is the answer. During the processing of hnRNA, capping and tailing, both of the processes, capping is done at 5' end and at 5' end there is addition of methylated guanosine triphosphate. So answer is methylated guanosine triphosphate is added to 5' end of hnRNA. A codon is said to be degenerate. Now what is degeneracy of a codon? Degeneracy means when the same amino acid have more than one codon, that is degeneracy. exception of degeneracy of codon is methionine. The methionine have only one codon, that is AUG, which is the start codon. So tryptophan also is the exception for this property. And next question is, a nucleoside is formed through N-glycosidic linkage by joining. A nucleoside is a pentose sugar and nitrogenous bases. So this bonding is N-glycosidic linkage. So the answer is nitrogenous base and pentose sugar. In eukaryotes, DNA polymerase 3 transcribed tRNA, 5sRNA and snRNA. This is very important function of all three types of RNA, very important for NEET. If you haven't done it, please refer NCERT for functioning of all three types of RNA polymerases. According to HGP, the Y chromosome in human being have 231 chromosome, while the it is the smallest chromosome, and chromosome number 1 is the largest chromosome and have 2968 genes. In prokaryotic translation, the formation of peptide bond between two amino acids is catalyzed by the answer is ribosome. The actual structure of tRNA. If we consider, it is inverted L shaped. The technique of DNA fingerprinting involves satellite DNA and it acts as probe, and this satellite DNA have a very high degree of polymorphism. That is why we use this in this process. So the answer is A. What is the role of tRNA in protein synthesis? tRNA binds to specific amino acid. It acts as transfer RNA. It brings amino acid to the complex for synthesis of protein. In lac operon model, the role of lactose act as inducer molecule. If lactose is present, in the presence of lactose, repressor protein is getting inactivated. Lactose actually binds to repressor protein, and hence repressor protein do not bind to operator region, and transcription and translation, both the processes take place. So in lac operon, lactose acts as inducer and by activating the repressor. That statement is correct. So next is promoter terminator. Promoter is RNA polymerase bind to initiate transcription. It initiates transcription, and terminator is end of transcription. Terminator represent end of transcription. Genetic code is triplet of nucleotide, and start codon, that is methionine. So answer is 2314. B. Next is statement 1. The I gene of lac operon codes for repressor protein. If you consider the structure of lac operon, just see this. I gene, then P, O, lac Z, Y, lac Y and lac A. Right? That is the structure of lac operon. The I gene is repressor regulatory gene which produces mRNA and then repressor protein. This repressor protein binds to the operator region for inactivation of lac operon or switching off of the lac operon. So the I gene of lac operon code for repressor protein. That statement is correct. In the absence of lactose, suppose if the inducer is present, it binds to repressor protein, and if lactose is present, it will not bind to operator region, and the lac operon is switched off. But if lactose is absent, suppose lactose is absent, then it will bind to operator region. So in the absence of lactose, the repressor protein binds to the promoter region, it binds to operator region. So this statement is incorrect. Statement 1 is correct, but statement 2 is incorrect. The commonly used host in HGP, there are two commonly used hosts, that is bacteria and yeast. Sevra-Okai enzyme is polynucleotide phosphorylase. This is the name of the enzyme. In an mRNA, untranslated region UTR are present. If we consider the structure of mRNA, so first we have a cap, then we have untranslated region. First we have a cap, then untranslated region, structural gene in which we have start codon gene and then stop codon. Stop codon can be 3 codons. Then again we have UTR and poly A tail. That is the structure of mRNA, right? So at 5 prime end we have UTR but before the start codon, and at 3 prime end we have UTR after the stop codon. So in an mRNA, untranslated region or UTR are present just after the start codon, incorrect. Just before the stop codon, incorrect. Only at 5 prime end, that is also incorrect. At both the ends it is present at both the end, that is 5 prime end and 3 prime end, that is absolutely correct. Read the following statement and choose how many are correct. Intron is a part of hnRNA, correct. Which does not code for any protein, that is correct. That is why hnRNA is inactive RNA. Regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Now this statement is very important. Now what does it mean, regulation of lac operon can also be visualized? If we consider functioning of lac operon, so here we have already discussed, let's see, functioning of lac operon. In this condition lac operon is switched off, means DNA is not transcribed into RNA and RNA does not make any protein like Z, Y and A. All three cistrons are not expressing its character. But if lactose is present, then all three genes will get expressed. Like Z will code for beta galactosidase, Y permeases and A transacetylases. All the three enzymes work together to perform metabolism of lactose. So basically lactose is the substrate which regulate the switching on and off of the operon system. So regulation of lac operon can be visualized as regulation of enzyme synthesized by its own substrate, and that is why the statement is correct. At the end of translation, a release factor binds to stop codon. That is also correct. An adapter molecule would on one hand read the code and on the other hand binds to specific amino acid. Adapter molecule means tRNA, and that is the function of tRNA, that at one end it reads the codon through anticodon loop, and on the other hand amino acid getting attached to the tRNA. So all the statements are correct. Match the column. Griffith's experiment, transformation in bacteria, we have already discussed this experiment. Jacob-Monod and lac operon. Hershey and Chase, they gave unequivocal proof that DNA is a genetic material. Michelson and Stahl experiment proved semi-conservative replication of DNA. They used heavy and light nitrogen. So answer is A. The size of variable number V and T are variable number of tandem repeat is mini satellite, and the size of mini satellite is 0.1 to 20 kb. DNA used as genetic material as well as catalyst, that is correct. Primarily it is act as messenger RNA, but RNA can be used as genetic material in some viruses, and it can also act as a catalyst. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by any protein enzyme. Why RNA act as catalyst? Because there are some important biochemical reaction in living system that can be catalyzed by RNA catalyst and not by any. This enzyme is ribozyme, which is made up of RNA. Why these reactions are catalyzed? Because RNA can also act as catalyst. So if we switch the statements, the answer will be A, but according to this question, the answer is B. So there are some biochemical reaction in living system that are catalyzed by RNA. Why it is catalyzed by RNA? Because RNA can also act as catalyst. So answer is B. If we switch the statement, answer will be A. RNA polymerase associate transcription with initiation factor sigma to initiate the transcription, correct, and rho for termination. Splicing is the process of removal of introns and joining of exons in a defined manner, that is also correct. That is the process of splicing. A messenger RNA is 336 nucleotide long, including the initiator, okay, and terminator codon, the number of amino acid in the protein translated from this mRNA. If we consider there are 336 nucleotide, then divided by 3, is the codons, right? There are 1, 1 to 112 codons, out of which one is stop codon, which do not code for any amino acid. So how many amino acids are there in the protein chain? 111, including start codon, but stop codon do not code for any amino acid, so the answer is 111. Next is statement 1, DNA polymerase catalyze polymerization in one direction, that is 5 prime to 3 prime, that is correct. During transcription of DNA on one strand, during transcription of DNA on one strand, the transcription is continuous. This is actually the statement for replication and not transcription. So statement 1 is correct and statement 2 is incorrect. Next is HGB was closely associated with rapid development of new area in biology, that is bioinformatics, that is correct. Capping and tailing of heterogeneous nuclear RNA means H and RNA take place at 3 prime end and 5 prime end respectively. Capping take place in 5 prime end, tailing at 3 prime end. So this statement is incorrect. And amino acid sequence in protein is synthesis is determined by the sequence of mRNA, that is absolutely correct. The sequence of mRNA determines the amino acid sequence. Enzyme catalyzing synthesis of RNA over DNA template is DNA dependent RNA polymerase, that is also correct. This is the enzyme used in transcription. Next is DNA replication is semi-conservative, correct. DNA polymerase add nucleotide in 3 prime to 5 prime direction, that is incorrect because it should be 5 prime to 3 prime. Polymerization direction is 5 prime to 3 prime. Replication is bidirectional in prokaryote, this is correct. Replication is semi-conservative, bidirectional. Replication occur in S phase of the cell cycle, this also we have discussed. So answer is A, C and D are correct and B is incorrect. Which of the following statements are correct regarding the leading strand during replication? It is synthesized continuously, correct. Polarity of template for leading strand is 3 prime to 5 prime, or polarity of template for leading strand is 5 prime to 3 prime. If we consider leading strand, so polymerization is obviously 5 prime to 3 prime, so the template for that strand should be 3 prime to 5 prime and not 5 prime to 3 prime. So statement 2 is correct, 3 is incorrect. It is synthesized in the same direction as replication fork, so this is the replication fork, this is leading strand, this is continuous synthesis. So the direction of polymerization is toward the replication fork. So that statement is also correct. Answer is C. Only A, B and D are correct. C is incorrect. Which of the following statement are correct regarding lac operon? Lac operon is an example of inducible operon. That is correct because it is regulated by inducer. And the lac operon is regulated by repressor protein. If it is regulated by repressor protein, then it is negative regulation. This is also correct. It is found in prokaryotes such as E. coli. That is correct. It is found in prokaryotic cell. Lac operon system is not found in eukaryotes or human. Lactose acts as inducer for the operon. That is also correct. And we have already discussed the structure of operon. So all the statements are correct. Next, translation is formation of polypeptide chain, means protein. Transcription is formation of RNA. Initiation of translation is start codon, that is AUG. Post-transcriptional modification is splicing, which is in first option. So the answer is B. Next is ribosome. Ribosome acts as site of protein synthesis. tRNA brings amino acid to the ribosome. mRNA has information for polypeptide synthesis, and RNA polymerase 1 catalyze the synthesis of RNA. So the answer is C. Why did Hershey and Chase experiment use bacteriophage? Hershey and Chase gave unequivocal proof that DNA is the genetic material, and they use bacteriophage, that is a virus which infect bacterial cell. So bacteriophage, basically the genetic material is double-stranded DNA, and it also contain a capsid protein coat. So bacteriophage contain DNA and protein coat. So we have to find out that what is the exact genetic material, either DNA or protein. So they use bacteriophage as the model organism for their experiment. What was the final conclusion of Hershey and Chase experiment? Hershey and Chase experiment proved that DNA is the hereditary material and not the protein, because that was the confusion that time, that either DNA or protein is the genetic material. So they explained that they gave unequivocal proof that DNA is the genetic material. A gene encoding a polypeptide of 50 amino acid get mutated at 25th codon, UAU becoming UAA, which is a stop codon. So at 25th amino acid, stop codon is added. So 24 amino acids are formed uninterrupted. There are no disturbance. But at 25th codon, the process of translation get interrupted, and since it is a stop codon, it will terminate the process. So polypeptide of 24 amino acids are formed, and here the process of translation stops. Histones are rich with amino acid. Histones are rich with amino acid lysine and arginine. Which of the following is or are correct statements with reference to RNA? RNA is more reactive than DNA. That is why it is not the genetic material. It is less stable than DNA. It mutates at a faster rate. It mutates at a slower rate is incorrect. So the answer is B. A, B and C are correct. The unequivocal proof that DNA is the genetic material came from, we have discussed it many times in this paper, that Hershey and Chase gave unequivocal proof that DNA is the genetic material. Which one of the following is not an important goal of HGP? If we consider goal of HGP, then determining the sequence of 3 billion base pair is the goal, storing the information is the goal, improve the tools for data analysis is also the goal. But 10,000 to 15,000 gene, this data is incorrect. So the answer is A. Now, arrange the following DNA sequences in ascending order of the number of hydrogen bond. If we consider hydrogen bond, then in option A, we have 1, 2, 3, 4, 5. 5 guanine and cytosine means 5 into 3, 15 hydrogen bond, and 1, 2, 3, 3 adenine and thymine, that means 6 hydrogen bonds. So it makes 21. Correct? In this 1, 2, 3, 4 means 8 plus 1, 2, 3, 4. 4 into 3, 12. This is 20. 1, 2, 3, 4, 5. 5 into 2, 10. And 1, 2, 3 plus 9, 19. Similarly, in this 18 hydrogen bonds are there. So the sequence is very clear: A, B, C and D. So the answer is this. In a DNA molecule, GC rich part melt before the AT. AT rich have only 2 hydrogen bonds. These are 3 hydrogen, which is stronger region. So this statement is incorrect. Between A and D there are 2 hydrogen bonds, correct, and between G and C there are 3 hydrogen, that is also correct. So the statement as a reason is correct, assertion is not correct. Answer is C. VNTR becomes a very useful identification tool in forensic application, that is correct. And the reason for this is because VNTR from every tissue from an individual shows the same degree of polymorphism, whether we take the sample from blood or saliva, hair, any sample we take, it shows same degree of polymorphism. That is why it is used an important tool for forensic application. So answer is D. It is the correct explanation. Next is which one represent the correct manner of DNA replication. This is the correct answer: leading strand, lagging strand. right? So 3 prime and 5 prime end for leading, and 5 prime to 3 prime for lagging. It is toward the fork, it is away from the fork. Next is spliceosome are not found in cells of. If we consider spliceosome, then spliceosomes are formed in the process of splicing, and splicing process is characteristic feature of eukaryotes. So in streptococcus bacteria, splicing is not performed. So the answer is streptococcus. DNA replicates semi-conservatively. Which of the following scientists perform the experimental proof for this? The answer is Michelson and Stahl in prokaryotes and Taylor in eukaryotes. So both are correct. Diameter of DNA is 20 angstrom. Distance between two adjacent base pair is 3.4 angstrom. The pitch of the helix is 3.4 nanometer. The base pair in nucleosome is 200. So the answer will be B, 2413. What is another chemical name of thymine? Thymine is 5-methyl-uracil. In prokaryotes, hnRNA is subjected to a process called splicing, capping and tailing. In prokaryotes, processing of hnRNA is not done. This is incorrect. Reason is, in eukaryote, the primary transcript hnRNA is non-functional. This statement is actually correct. So assertion is actually false, but reason is true. UTR, untranslated region, is present in both 5' and 3' end in the mRNA. This is correct. In eukaryotes, the primary transcript called hnRNA contain both exons and introns, and both are functional. Both exons and introns are not functional. So statement 1 is correct, but 2 is incorrect. Which one of the following is the correct statement? Cesium chloride is used in Michelson and Stahl experiment. This is correct. NH4Cl is not used in Griffith's experiment. This is also not used in Hershey and Chase experiment. It was used in Michelson and Stahl experiment. Radioisotope used in Griffith's experiment. It is not used in Griffith's experiment. It was used in Hershey and Chase experiment. So the answer is A. If there are 999 bases in an RNA that codes for a protein with 333 amino acid, and the base at position 901 is deleted, so there is mutation at 901 nitrogenous base, such that the length of the RNA becomes 998 bases. How many codons? So after 999 nucleotides are there which code for 33 amino acids, correct? So the mutation is at this position, so almost 33 amino acids are altered. In Frederick Griffith's experiment, what happened when heat killed S bacteria along with R bacteria injected into mice? When heat killed S strain bacteria along with R strain bacteria was injected into mice actually died, because the DNA from this bacteria get entered into this R strain bacteria by the process of transformation. So R strain bacteria get transformed into S strain bacteria, and that live S strain bacteria was extracted from dead mice. So answer is B. Mice died and showed live S strain bacteria. Next is lac-operon consists of 1 regulatory and 3 structural genes, which is Z, Y and A. Genes Z, Y and A are codes respectively for answer is this: beta-galactosidase, permeases and transacylases. A template DNA strand is given as this is the sequence given for template strand. We have to find the sequence for RNA transcribed from above DNA. So if the template strand is 3' to 5', then the polarity of RNA is 5' to 3'. And in RNA, thymine is not present, uracil is there. So the sequence is this: adenine, complementary of adenine is uracil, thymine, adenine, guanine, cytosine, guanine, adenine, uracil, and similarly this is the answer. Blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences is termed as sequence annotation and not express sequence time. Many non-human model organisms such as Cinerobius reptitis elegans, a free-living non-pathogenic nematode and not pathogenic nematode. So this is also incorrect. So both statement 1 and statement 2 are incorrect. Which of the following are true regarding the role of lactose in lacoperon? Lactose is an inducer which binds to repressor protein and inactivates it. So lactose bind to repressor and inactivated, correct. Lactose ensure replication of structural gene, incorrect. Lactose directly bind to the promoter, incorrect. Promoter is binding site for lactose only, that is also incorrect. So true is only A. Which of the following statements are incorrect about lacoperon? The structural gene code for enzyme involved in lactose metabolism, that is correct. Beta galactosidase, permeases and transacetylase, all the three enzymes work together to help in lactose metabolism. The promoter is the binding site for repressor. It is incorrect. It is the operator region actually. And in absence of lactose, structural genes are not transcribed, that is correct. And lack A gene code for transacetylase, that is also correct. So incorrect is B. Statement 1, more than 6%, it is less than 2%. Genome code for any protein, so this is incorrect. Repeated sequence make up a very large portion of the human genome, not a small portion. So both the statements are incorrect. Answer is A. In eukaryotes, gene regulation can be exerted at the following level. It is transcriptional level, number 1, then processing level, number 2, and transport of mRNA, number 3, and this is 4th. So answer is B, C, A, D. C. Human Genome Project was launched in the year 1990 and ended in 2003. In E. coli, the lac operon was switched on. Lac operon was switched on when repressor binds to operator, that is switched off condition. RNA polymerase bind to operator, that is also incorrect. Lactose is present and binds to I gene product. I gene product means repressor protein. So it gets inactivated, due to which lac operon gets switched on. Read the following two statements and select the correct option. During DNA replication, deoxyribonucleoside triphosphate serve as dual purpose, that is correct. It provides energy and act as substrate. E. coli that has only this much base pair completes the process of replication within 18 minutes, that is also correct. Both statement 1 and 2 are correct. Phosphoric acid remain associated with which of the following carbon of sugar in nucleotide? So the answer is 5th. If we consider pentose sugar, so first carbon is for nitrogenous base and here is the 5th carbon which is for phosphate group. Select an option that shows the correct sequence of event involved in translation mechanism. So if we consider the sequence, then charging of tRNA is the first event, then binding of mRNA to the smaller subunit ribosome is the second event, then binding of initiator tRNA to the P site is the third event, then formation of peptide bond between amino acid present in P and A site is the fourth, and formation of further polypeptide chain is the last step. Number of genes associated with chromosome 1 in human being is 2968. Mark the correct match with column 1 and column 2. 6.6 into 10 raised to the power 9 diploid set of human DNA. 48502 base pair is lambda phage. 5386 is 5 1 and 4, and 4.6 that is for E. coli. So 4312 is the answer. Who for the first time pointed out that triplet of codon? That answer for this question is George Gamow. Who provides stability to the helical structure of DNA? There are two reasons: hydrogen bonds and staking of bases one over the another is the other reason. There is a crime at Jantia Hills. Dr. Ansari, who is a forensic uh, specialist, collected DNA from the crime scene and suspected two individuals, that is Ramesh and Anurag. He analyzed the different copy number of VNTR. He used chromosome number 7, 2 and 16. After his observation, he cleared that the banding pattern of DNA matched with Anurag and not with Ramesh. That means who is the criminal according to Dr. Ansari? That is Anurag. The fact that a purine base always pairs through hydrogen bond with a pyrimidine base leads to which feature in the DNA helix? Is uniform width throughout the DNA. If we consider purine, then purine is double cyclic ring and pyrimidine is single cyclic ring, which maintains the width of the DNA, constant width of the DNA, that is 20 angstrom. In Michelson and Stahl experiment is continued for 4 generations in bacteria. The ratio of heavy nitrogen will be 0 after 4 generations. In any generation, DNA with heavy nitrogen, completely heavy nitrogen is 0. Hybrid is always 2 and light. After 4 generation, how many DNA are there? 2, 4, 8 and 16. Out of 16, 2 are hybrid, then 14 will be light. So the ratio will be 0, 1 and 7. The base pairing confers a very unique property of the polynucleotide chain. That is correct, because they are said to be complementary to each other, and therefore if the sequences of one strand is known, then other strand can be predicted. That is the correct explanation. Next is the diagram for Hershey and Chase experiment, bacteriophage and E. coli. And next is the sensitivity of DNA fingerprinting technique has been increased by use of PCR. That is correct. The bulk DNA form a major peak and the other smaller peak are refers to as satellite DNA. Both the statements are correct. VNT belongs to a class of satellite DNA referred to as mini satellite. This statement is correct. Ribosome consists of structural RNA and 80 different type of proteins. Both the statements are correct. When the larger ribosomal subunit encounter the mRNA, it is incorrect. It should be smaller subunit. The formation of peptide bond between amino acid attached to the adjacent tRNA does not require energy. It is energy requiring process. So both the statements are incorrect. Presence of catalyst such as 23S RNA in bacteria, that is ribozyme, would enhance the rate of polypeptide bond formation. This statement is absolutely correct. A classical example of point mutation is sickle cell anemia and not thalassemia. It is an example of frame shift mutation. So answer is B. So I hope all the questions are clear. If you still have any doubt, then you can go to your subject teacher. Thank you.